Thank you so much, Warren, for kind introduction. I'm very happy to come back here. I attended the first meeting 2 years back, and I feel very fortunate to meet you guys and share my research with you. So as Warren mentioned, I'm from Dr. Goldstein lab. He talked before lunch. So we are mainly GI lab. Now we work on enteric nervous system. So before I go to the main talk, I'll give you some introduction about the GI physiology. So you all will agree with me that our well-being heavily depends on the gut function. If the gut works well, it helps in digesting food, like getting energy from the food, removing waste material, and even controlling our mood. But gut functions depend on few of the components. Like I mentioned here, a few of them, like the immune system, enteric nervous system, small muscle cells, microbes, they all crosstalk each other. If the crosstalk, if their brilliant dialogue works well, then the gut function goes well, and so we do. So I'll walk you through very briefly on like the few of the systems. Like I'll talk first about the gut immune system and the microbes. So you see the this is a cartoon. Do you have the
right?
Yeah. So in the cartoon you see the intestinal lumen side, the hollow pipe, the internal side. That's called like the first layer is mucus. Then you have submucosa. Then you have two muscle layers. In the muscle layers we have the neurons. I'll talk about details about the neurons later. This is a HA staining picture again from very bottom. These are top or bottom, whatever you say, from here to all the way here is the mucosa. When we eat food, all the food materials during the digestion, it comes in contact with this layer. Our food contains lots of microorganisms. So these are the immune system. Most of the immune cells are residing in these layers. It's called mucosa, like the mucosa layer. All types of immune cells are mostly there to safeguard our gut system. Then we have the submucosa. Submucosa has got few immune cells as well. And muscularis and the serosa, the outer surface, they have very less immune cells. Immune cells should not, immune microorganisms, pathogenic bacteria should not go to that layer. It should be mostly at the surface in the mucus layer. So we have the immune cells mostly in those layers. Then the smooth muscles, that's like the whole purpose of this conference. So that smooth muscles, they actually like the contract, relax with his own wish. So you know, like when the calcium comes inside the smooth muscle cells, they start contracting through this pathway, like the calcium, free calcium that actually activates the calmodulin dependent like the myosin light chain kinase that activates the phosphorylates the myosin chain to cross-bridge with the actin, and finally we get the muscle contraction. But this muscle contraction is very different from the other organs. Like in the gut, in different parts of the gut, starting from the esophagus, stomach, small intestine, large intestine, their function is a bit different. They all contract, but the purpose of contraction is different in different places. Like if you think about the small intestine, most of the absorption happens here. The food kind that moves moves very slowly. They go forward, come backward, go forward, come backward to give more time to absorb the energy, nutrients from there. And in the large intestine, in the colon, when after happening most of the absorption, you have just the waste material to get rid of the body. There, the motility pattern is very different. That's why we are saying we listed few of the motility patterns: peristalsis, segmentation, migration, motor complex. So same smooth muscle cells in different places, they work differently. But these differences actually not sensed by the smooth muscle cells. They just do the contraction relaxation. That motility, that pattern is actually controlled or regulated by the enteric nervous system. That's our third, and the main component actually in the total gut. One of the main components is smooth muscle cells and enteric nervous system. What is enteric nervous system? Enteric nervous system is like in our body, other than the brain, other than the neurons in the spinal cord, this is a huge number of neurons sitting in the gut. That's why we call it second brain. There are more than 500 million neurons are there. They are controlling our gut function, mostly like the contraction, relaxation, that is motility, and also the absorption. These neurons are actually residing into different places. One is in the submucosa. After the mucosa you saw in the previous picture, there is a hollow space. That is submucosa. Those neurons are actually controlling our absorption from the food, or we absorb the nutrients. That is mostly controlled by the submucosal plexus neurons. Then we have got the neurons in the muscle layers. In between two muscle layers, we have two muscle layers. There's one is called circular muscle, another is longitudinal muscle. In between these two muscle layers, we have the other type of neurons that called myenteric neurons. They actually control the muscle function. In the bottom, you see a picture. It is continuously contracting. It is out of the body. It is a mouse colon. It is taking out of the mouse body. It is in a crepe solution. But you see it is working very with the harmony. You see from the top to bottom, it's not going other direction. It's going just in one direction. This is the enteric neurons function. They actually control these pattern and also these like the direction, everything. So other than these, like in the enteric neurons, we have in the myenteric plexus, we have mostly two types of neurons. One causes contraction. They are called excitatory neurons. The neurotransmitter, the chemicals what are released from that neurons are called excitatory neurotransmitters. Mostly acetylcholine and substance P. Acetylcholine is the main excitatory neurotransmitter that binds with the receptor sitting on the smooth muscle cells, and that ultimately cause contraction. Then we have some inhibitory neurons, some chemicals that actually cause relaxation to the smooth muscle cells, mostly nitrogen neurons. Nitrogen neurons actually bind with the that causes the cyclic GMP, ultimately to stop the contraction. In other way, you can say the relaxation when the contraction is stopped. So other than the enteric neurons, gut can also be controlled by the central nervous system. We know we have two hands of central nervous system. One is called sympathetic, other is parasympathetic. They're also coming to the gut to modify the enteric nervous system function. Enteric nervous system can work independently, but sometimes it is also controlled by the central nervous system. So again, the first slide I showed, they all need to talk very coherently. There should be very smart intellectual dialogue between them. If they work together, will be good. If anything goes wrong, then we see the lots of abnormalities, lots of side effects like the inflammation or different types of motility disorders. And sometimes the neurons can be impacted, sometimes the smooth muscles can be impacted, or immune system and other system. So these slides you have seen a few times today. I'm not spending time on these slides, not here as well. You see the gut dysmotility in the patients is already clinical symptoms of these patients. They have gut dysmotility, malrotation, these type of things. So we are very fortunate that Dr. Mussolino, Dr. Lindsay, they have been very successful to create the mice model. So this is the mice model. You see the abnormality of this mouse from the healthy to the disease. This is the smooth muscle cell. They have shown that the alpha actin, that thing is mutated, and they are not the structure is not homogeneous in these groups. So this slide was also task based by Dr. Mussolino. So I'm going here. So we are interested in the GI physiology. So we look at the gut. You see the difference here. The control gut, it is taken out of the body. It is in a petri dish. It is moving, rotating continuously. Look at the other mutant group. They're very silent. They're not much things. Like no brainer. Anyone can tell what's happening here. So we looked at first we looked at the structure of this tissue, of this colon. We measured the length of the small bowel and the large bowel. The small bowel is starting from stomach to all the way cecum. That part is mostly responsible for the absorption, getting energy from the food. You see they become shorter in the mutant mice. Then we have the large bowel. It is starting from cecum to all the way rectum. You see they become shorter in this in the mutant group. And you see also the difference. The cecum is dilated in this group, and the fecal fillet is, you see, they are very different from the normal shape of the fecal fillet. They are dilated in the proximal part, and in the distal part, they are actually constricted. That's why all these mice, we open up the tummy, we see there is a big chunk of like the waste material deposited here. They're not coming out of the body. That can be one of the reasons of these mice to die early. Then we did the histology to see the other structure. We did not go more deeper. We just measured the width of the muscle layer and the mucus layer. You see the muscle layer is thinner in these. When we see these mice, many times this outer surface is very, very thin. And sometimes I found one or two, they are perforated because these fecal fillet is just sitting there and the muscle becomes thinner, so it perforates sometimes, and the mucosal layer becomes thicker in this group. Then we did something called colonic migration motility complex. Yeah, I'll turn the video on. You will see it is contracting. If you count this contraction, you will see that contraction. That 10 minutes video was converted to a thymograph. This map, you see the map, this red is indication of the contraction. If you count contraction, you will see that many times colon is contracting, and it is also superimposed with the interluminal pressure. We measured the pressure. So when this contraction happens here, you see this red line that is created by the software, but we can see at the same time change in the interluminal pressure. That means your colon is closed at the top and at the bottom. As the liquid cannot escape, when this contraction happens, this pressure goes up. We measure that pressure and you see it's superimposed. Every time there is contraction, we can see the change in luminal pressure and we can see this band of contraction. And if I go to the mutant group, you see they are shaking, you see they are vibrating, but there is no beneficial movement from left side to right side, from oral to anal side. They do not go away. It's just vibrating there. You see you see the map. We don't see that. You see it is red. As I said, red is the contraction. You see red is all over the places. They're vibrating, they're contracting, but that contraction is not beneficial. That is not moving from oral to anal. We measure the luminal pressure. Again, it is very low there. Okay, here I'm showing you something. We have in Dr. Goldstein's lab, we are very very focused on one particular disease that is called Hirschsprung disease. You might have some of the parents of the kids, you might have heard this term as well, because when your kids have started this, when you start noticing something is different in your kids, one of the symptoms you can see their belly is like the dilating. You can see the dilated belly. Last year I talked with Dixie, and she was saying that her child was first diagnosed. Doctor they thought it is a Hirschsprung disease because her belly is enlarging and she is not pooping. So we have in our lab, we have a model that we call like the mimic that disease, Hirschsprung disease. We have a mouse model is called EDNRB knockout. That mouse had got the very similar. Anyway, that video is not running. Very similar pattern of vibration or contraction. There is no actual contraction there. Anyway, we measured those parameters, like how many times they contract in 10 minutes. This contraction is different. They're called colonic migration motor complex. Usually 6 to 7 times in the healthy mouse, but in the mutant you see 1 or 2 times. And we measured the velocity. How long does it take from move from oral to anal end? Is around. You see, they all are like they're different in the mutant group. Then we measured, looked more in other parameters. One of the parameters, we looked at the total GI transit time. The video I showed you, that is ex vivo. We took the gut out of the body, we put in the petri dish. But when in the body, when your whole system is working together to see what is actually gut doing. So we put some red dye in their stomach, and we wait 4 to 8 hours to see when the red poop comes out, because that dye does not absorb. So that is our total GI transit time. You see, it is around 3 to 4 hours in the control mice, but in the mutant, it takes 6, 7, 8, 9 hours to get the first red fecal pellet out of the body. We also looked at the X-ray. We did these X-ray. We put the liquid, and we also put the solid. And you see, there is no difference in the gastric emptying. Surprisingly, their stomach looks normal. It is not different in the control and the mutant group. Then we looked at the rectal bead expulsion. As this thing is delayed or prolonged, somewhere is problem, not in the stomach, then where it is. We looked at the very rectal part. We put a glass bead, we inserted around 2 to 3 centimeters. We took the time, how long does it take to come out? That glass beads come out very quickly in the control mice, but that mutant mice took time to come out. It is delayed. That means they have mainly problem in that narrow constricted part. I showed you the picture before. It takes, due to the narrowness actually is the main cause of all these things. To look at the mechanism why it is happening, to look more closely, we did the smooth muscle contractility test. We put a small piece of gut in the organ bath. We measured their contraction. You see the pattern of the healthy mouse. They have like these small spikes of contraction, and suddenly when we give the electrical field stimulation, we see a big contraction. What does electrical field stimulation do? Electrical field stimulation actually excites the enteric neurons. I talked before, particularly both the excitatory and inhibitory neurons, they release their neurotransmitters that bind with the smooth muscle receptors, and ultimately smooth muscle will contract and relax. You see here, after giving the stimulus, we see a big contraction because neurons release the neurotransmitters, bound with the receptors, and we see a big contraction. But look at here. In these mutant mice, they have a pattern, they have a rhythm of smooth muscle contraction. It's not like unorganized in the control mice. They have a very rhythmic pattern. Then we give the electrical stimulus. If I don't say that I did not give electrical stimulus here, you will see everything is safe. That means it does not do anything. We measured that maximum contraction. We see the actor, that mutant group has got, actually did not do anything. You see, it should be zero actually, but some of the mice, we see little bit of contraction, and this is just one random example. It does not do anything. What's happening? Anyway, what is this?
I think someone's writing on the screen.
Someone is writing, or
That's a cool trick.
someone is controlling us.
All right, Jane again.
Okay, cool. Now I have no control. Okay, beautiful. So you see, this is the what is called. You see here, I just showed you the healthy mouse. We have this type of pattern. Look here. So I put not one trace. I put you few of the traces. You see the pattern of the control mouse, healthy mouse, is like this. You cannot say anything. They don't have any pattern. Each of the mouse, they have different types of contraction. This contraction is actually mediated by the enteric neurons. So how the sensory neurons sense what type of contraction they need, they just produce that type of contraction. But in the ACTA2 mutant mice, they don't have any crosstalk with the enteric neuron. Enteric neurons may not take the control. That's how the smooth muscle cells, they are doing their contraction in a very rhythmic way. You see that you can see a pattern every time they are doing at a regular fashion, not in the control healthy mouse. Then as I give you, I tried to talk about the Hirschsprung mouse, that's the Indian Army knockout mouse, you see, they're exactly the same. They have a pattern. They have a pattern. All of these are from recording from different mouse. So these actually give us the impression that these mouse can be model of Hirschsprung mouse by mistake. But what is actually Hirschsprung mouse? They don't have enteric neuron. I should actually give you a little bit of introduction about the Hirschsprung disease. The baby borns without enteric neurons at their very rectal part. That's one type, and sometimes segmental. In different portion, they have no enteric neurons. Baby without enteric neurons at the very rectal side, they cannot poop. That can be one of the reasons if you do not do the treatment, they will die. So we thought it is that model that they might not have enteric neurons. To check this thing, as our electrical field stimulation was supposed to release the neurotransmitter, if the enteric neurons are damaged, we cannot get that thing. So we are giving acetylcholine from outside in the bath. You see what happens in the control mice, in the low dose, little bit of response or no response. When you come to the high dose, we see a big, like the contraction, more frequent, big contraction due to the acetylcholine. That's the normal. It should be the case. It should happen. But look at the mutant mice. When we give the acetylcholine, it should at least, you see, if it does not do anything, it should stay the same. But actually, they lose their contraction. That means they can become hypercontractile, or anyway, they are just losing that pattern even with the acetylcholine. I'll give you these. I'll explain this little later. But look at here that the acetylcholine does not do anything in the actor to mice. Sometimes it even inhibits their pattern of contraction. Then we tried with the KCL, that's called the global depolarization. Again, it shows the abnormality of these actor 2 mutant mice. Again, we added other thing that's called tetrodotoxin. That's a toxic substance. If we add this thing, what does it actually do? It stops the communication between the enteric neurons and the smooth muscle cells. So we can mimic it with this chemical as Hirschsprung disease. Hirschsprung disease, they don't have the enteric neurons. They cannot cross talk with the smooth muscle cells. We are doing that thing. We are adding the TTX. So what does that mean? The neuron is not controlling your smooth muscle anymore. Then you can see high magnitude, high frequent contraction. Why? Because your enteric neurons actually mostly controlled by most of the time with the inhibitory neurons. So when those inhibitory neurons are stopped, you see the muscle is doing it with its own pace, however they want. That's their pattern, and exactly that is same in the actor 2 mutant mice. So again, it is giving us some hints that something can wrong with the neuron, or neuron can be there, they do not cross-talk properly with the smooth muscle cells. So then we counted the total number of neurons. Is really any change in the neuron number. But you see that this is the control wild-type mouse. They have beautiful, U is the pan-neuronal marker. These are the enteric neurons. We looked at other subclass that's called NOS. We looked other subclass that called calritenin. So you see all the neurons are same. There is no change in this mouse, mutant mouse, compared to the healthy mouse. So that indicates that the enteric nervous system is excellent in these mutant mice, but the problem is they cannot crosstalk with the muscle. To find out further mechanism, we did the single nuclei RNA sequence. It is little different from single cell RNA sequence. So here we take the colon from the control mouse, from the mutant mouse. We took the mucus layer, submucous layer, only the muscle layers are there, and also the enteric neurons are there. First, we looked at whether we can capture all different types of cells in our single nuclear RNA sequence data. You can see all the different types of cells. What should be present there? They are present in both control and the mutant mice. You see that we use 2 markers for smooth muscle cells. These are smooth muscle cells with the MYH11, with the ACTA. You see the mesothelial cells, glial cells, neuron, all the types, appropriate types of cells are present there. So our method is validated through that data. Then we look the differential gene expression analysis to see what types of cells are more affected in this sequence data. We see in the mutant group, the smooth muscle cells came out the number one, like the types of cells, they are affected in these mutant mice as expected. Other type of cells, neuron, fibroblast, most of the other types of cells are no difference between these ACTA2 and the healthy mouse. We see little bit of difference in the mesothelial cell. We can explain that thing in the proximal colon. We see dilation, we see the stretch, that's how the mesothelial cell can be impacted, but it is mostly smooth muscle cells. Then we interested to see what types of genes are changed in these smooth muscle cells. We find around 200 types of genes are upregulated in smooth muscle cells, and 120 types of genes are downregulated in smooth muscle cells of the mutant mouse. And we see some of the genes like the PDE10A, Parkin, they are very upregulated in the mutant smooth muscle cells. What does this gene do? It is actually the marker for the enzyme what actually degrade the cyclic GMP. If you can remember, I told you, I'll tell you later about that what is called, they're getting tired or whatever, acetylcholine, or they have like the rhythmic contraction, or acetylcholine does opposite thing. We believe this gene can be one of the things that causes the breakdown of cyclic GMP. Cyclic GMP causes the relaxation of your muscle. So your relaxation apparatus is now damaged. What you will see? You will see only contraction. That's what we see in this mouse. They have contraction that is very abnormal. We don't want that contraction. We want they should be regulated by the enteric neurons. Anyway, moving forward, then we look at these types of genes. We did the gene ontology analysis of those differentially expressed genes. We have seen they are related with the actin binding and actin cytoskeleton. These differentiated genes are actually associated with actin binding and actin cytoskeleton. So from here, I get the confidence that we see mostly the abnormalities, the actin gene and also the cytoskeleton. That's how my big answer to the question. When neurons are there, why neurons cannot talk to the smooth muscle cells? The receptor may not be sitting how it should be. When we talk about the receptor in our body and the drug, we use one word, lock and key model. How key should be matched with the lock, your receptor should be very matched with the agonist. Acetylcholine will be released. They have the cholinergic neurons there, but why they are not impacting the smooth muscle contraction relaxation? Maybe they have the receptors, but they are not sitting in a shape that can be twisted, that can be altered. So that's the characterization. It is very well characterized mouse model that it talks what we want actually. The smooth muscle are abnormal. Now it's time to see the therapeutic benefit if we can change this. So first we'll start with sapropterin. Dr. Mussolino talked about in the morning, and he referred me that I'll refer back to her. She already showed. I'll show you a little bit, not much. So first, Vijay, he's here. He did the survival curve after the sapropterin treatment. Sapropterin is a small molecule already FDA approved. You see in these, I'm running late of time. Should I continue?
Yeah.
I'll be very quick.
Yeah, you're fine. Yeah.
Okay, I'm sorry.
We want to see it for sure.
Oh, yeah, yeah.
Can you go back?
Okay, so from the natural history curve, you see these mice stay live longer. So we looked at the gut again. We have actually, to be very honest, we tried different route of administration. We did give oral dose, we give like the IP sapropterin, we give high dose of sapropterin. So we see in the high dose of oral, there is little bit of like the what is called trend of getting normal. This is the structure. You see again, IP is something doing better in the large intestine. See, this is the mixed type of response. You see, when we did one of the mouse, I just randomly choose to see the colonic contraction. It is exactly as the normal. I'm not showing you all the video. You see, look at the band. They are contracting well. Their motility is well. What is called the luminal pressure is good enough. But some of the mice, I don't know how to explain these things. Some of the mice work beautifully, like the treatment works beautifully, turn it back to the very normal. Some does not work very well. If I combine these two sets of data, then I struggle to get the significance. But still, I got significance here with the oral HD suboptorin and like the luminal pressure, no change. With the velocity, I again see some change with the oral high dose suboptorin. So that's the drug or the dose. We can work more to see whether we can really we can really remove or like the improve the symptoms. We also did the smooth muscle contractility. I explained this thing before. Again, you see the oral HD, we see a little bit of the significance, although we have only 3 mice. So I would recommend Vijay to if you are interested to work on this drug, please continue. I'm happy to do the same thing what I'm doing. That's the suboptorin, and then we'll move into the gene editing. Miss first.
I haven't seen it.
Okay, so Miss Claire Fung, she presented very well. She gave the instruction of the construct, how the gene editing work, like what is the design. I'm not talking that thing. I'll just go directly to the response to the impact. You see the small intestine. I have only 4 mice to do this thing. The pattern is there, like in the actor 2 mice. You remember it was shortened, but we are just maybe reversing. It's early to say. Again, in the large intestine, same thing. The pattern shows that it does something. Then we looked at the gastrointestinal motility. As I said, like I'm not showing you the video again and again. This is the same video type of recording. You see in the actor 2 mice. So sorry, this is the GI transit time. If you remember, we give some dye, red dye. We wait for the fecal pellet come out. In the actor 2, it is very, very delayed. But again, in the GT group, I had only 3 mice to show that it is coming down. Rectal with expulsion, again, it is shorter than the control. I don't know what happened. But in the actor 2, it is delayed. That's what we don't want. It actually improved more what we did not actually expect anyway. But the thing is the gene editing is doing something. Then this is again a video to show that you see this is beautiful. Like this is like exactly the normal healthy mice. This is on gene treated mutant mice.
Wow.
It looks very normal, but again the question is why I do not get significant in all of them. Maybe some, I don't know, we have to work on that thing, whether the injection, when you are injecting that AAVPR with the very, very tiny small amounts, how efficient you are to do the injection. If you have some sort of marker to see at the end of the day after sucking the mice, okay, we'll analyze these mice because they have distributed very well, or they didn't, we'll pull out that data. Otherwise, we'll be just keep doing the experiment. When we add those type of negative and positive response, then we get nothing. So we have to come out with something. But confidently, as I'm doing experiment with like sometimes I'm blinded when the Dr. Mussolino's group give me mice, I'm blinded. I don't know whether it is what type of treatment. After giving the analysis, I give them data, then they decode, and it is beautiful. It sometimes comes out that the drug treatment is working. Then the EFS response, again, not significant, but you see the pattern that contraction is coming back after the EFS stimulus. Acetylcholine, again, the sensitivity is coming back. The pattern is there. I would not say it's significant yet. So we need to, you see that what I'm saying? You see, one is just here, and we all want sitting here, then we could have the significant, but we can do more. That's like I'm very grateful to Dr. Goldstein and other members in our lab. Before I go, I'll tell you something. In last November, I was invited to give a talk in Italy. This is called myopathy, visceral myopathy conference in Italy. This is fully organized by brilliant parents like you in Italy. These parents group are actually working very hard to get some sort of good news to support their kids. They are working, this is called visceral myopathy conference, but all the parents or all the research talk was on ACTG2. You know TG2, yeah. Like the gamma isomer of that actin, but they are very unfortunate. They even can't have a mouse model yet. How fortunate you are that you have the research is going very well. all the credit should go to Dr. Mussolino, Dr. Luze,
Don't say that.
Dr. Ben is not here. I see him, my salute to him to find so quickly to get some gene editing stuff to at least you can get some hope that one day is very near, you can have a treatment. I pray, I wish all the lovely kids become healthy. You are brave parents, my salute to you. Thank you. I'm happy to take questions. I'm though very late. I'm more than happy to take questions. Any questions? Yes.
Let's ask for first. Sure. There's already said or?
So it seems like the neurons, the neurons are there,
Yeah, 100%.
but they're not getting a feedback.
Yeah, they are not communicating.
So in the single nuclei and the neuronal expression, you see
There.
that there were
Beautiful.
maybe no changes
No.
between mutant.
Oh, my talk is, yeah, like they have removed. Can you go back, please? I can show you all the cell types. We had neuron, we have glia there.
Yeah, I saw it, but there's no differential
No
expression.
differential, no,
in the
no,
neuron.
no gene
You have to go,
difference.
you have to go back.
Anyway, that's okay. But I showed you the cell types to validate our nuclei, like the what is called like the sequencing data. We did that. We looked at all the cell types. They're present and no difference in the neuron and the glia, gene-wise.
Yeah, there's been some presentations now of single cell proteomics. So, I mean, it's kind of the next stage, and you don't see
Yeah, if you are interested.
mRNA
yeah.
differences, you could go after proteomics.
Ahmed,
Yep.
to your question of why some mice treated with suboptrain have effect and others don't, or why some gene therapy. For the gene therapy, we have the sequencing is right now happening at the moment
Yeah.
at Ben Klaus-Teiber's lab.
Yeah.
And that's gonna be one of the parameters. You'll
Okay.
we see really low
Yeah.
liver editing.
Yeah.
These mice have
Yeah.
more than 50% editing in the
Yeah.
liver.
Yeah.
If there is little liver editing, is
Yeah.
the question that did you get enough?
Yeah.
Or was the injection
Yeah.
successful? And then for the subopterin.
Yeah.
Is put in the water, and these mice, these are just taking as much water as they
Yeah.
want.
Yeah.
We're not
It's their
persecuting
choice,
them.
they eat, they drink or not.
And that's why Vijay tried the IP, the intraperitoneal, because then he said, "No, I'm gonna make sure that every single mouse gets the same dose every day." So I think there's always variability in the experimental stuff, and that's why you try to look
Yeah.
for that dispersion. And
But it's definitely, it does is doing something.
yes,
Yeah.
to me, the effect is clear, also
Yeah.
the survival
Yeah.
and everything. But I just wanted to say the data will come and we might be able to explain some of your questions too.
Thank you.